add my welcome to Young's very gracious welcome to all of you. So it's my pleasure to introduce the first of two keynote speakers. We have a wealth of wonderful people here. We have two great keynote speakers tonight, Bob Kingston, and tomorrow night, Geneviève Almouzny. So lots of great talks to come. So here we have a picture of Bob. His work, he's perfect to lead off because his work really spans the years of transcription right through to chromatin and epigenetics. Bob, I'm going to give you a little history of Bob. Bob, in his own words, he was born and raised in Boston. He was a faculty brat. His father was at MIT. He stuck around Boston and Cambridge. Let me just start out with that. He went to Harvard. He got his bachelor's there. He then had a little hiatus, went out to, thought he might see a little bit of the rest of the world, so he condescended to go out to Berkeley. He did his graduate work there with Mike Chamberlain and studied pausing of transcription, very prescient there. And then he came back, of course, to MIT, to Cambridge, did his postdoc work with Phil Sharp, yeah. There he worked on what many of us were working on at the time, using viruses to use as models for transcriptional regulation. He also studied c-Myc, again, very ahead of his time, studying its regulation of mutant c-Myc in HSP70 transcription. All right, so then Bob, again, not adventurous at all, decided to take a job at this lowly institution, Harvard Medical School and Massachusetts General Hospital, where he started in 1985 as an assistant professor, beginning his own lab, working on c-Myc and HSF in transcription, HSP70 transcription, again, another area that everyone was using as a model. He then went through the ranks and very quickly, professor in 1995. He is currently chief of the Department of Molecular Biology, has been since 2005 at MGH, and vice chair of the Department of Genetics since 2006. Before I get to his science, a couple more minutes. I want to just say that he's had many honors. He's really a great illustration of service to the community, where he's done lots of editorial work, lots of organizing of meetings, and he, has been elected to many illustrious societies, including most recently the National Academy of Sciences here in the United States. Very great honor to him. I want to mention that Bob is an amazing mentor. I really wanted to say this for all the trainees in the audience. Some of his previous trainees are here. He really is beloved by the people in his lab. You really feel that when you meet them and hear them talk about him. So I think it's really important to mention that. All right, so what is Bob known for? In his own lab, he's done amazing work on the structure and function of nucleosome regulatory proteins and machines, very interested in how these things are put together into complexes. Especially, he studied repression, doing really stunning structure function work on demonstrating activity of these machines in vitro, always digging for proof, always doing rigorous work on mechanism and the function of these regulatory complexes. All right, so rather than go through all of the work that he's done, I'll turn now to, well, first I want to mention one other thing about Bob, and one of the reasons we chose him for a keynote is that his talks are spectacular. They are you will see, they always are, and tonight will be no exception, if he ever gets to it, if I ever stop talking. They are a lesson in clarity, elegance, and exemplary logic you're going to see in 1 second. Okay, so I'm not going to let him off the hook. Okay, he's a serious scientist. You can see that. Here's a very serious picture of him as chair of molecular biology at Harvard. but beloved by students and colleagues for his amazing work in mentoring. But when you introduce a keynote, you have to give a very full and complete picture. So there's another side of Bob. Bob, as many of us know over the years, really enjoys a glass of wine, or maybe even more than that, you know. Here he is in Ken Zaret's immortal words. Ken made me swear I wouldn't say this, blowing off a session at a meeting. So this is Bob blowing off a session at a meeting. All right, so Bob, what? Ken took the picture. I will credit Ken with the picture. All right, well, so Bob has really grand visions. I've been explaining that over this introduction. So he and Ali Shalatafard discovered a very important establishment. Here, Bob is at Bob's. Bob's pub, and well, it's not exactly the Eagle. We have to mention that here at Cold Spring Harbor, the Eagle in Cambridge. But I'm sure Ali and Bob exchanged some very important ideas at that Bob's Bar. Okay, so now I promise you, we will have a talk from Bob. So Bob, it's a pleasure to welcome you and have you lead off our meeting.
Great. Thanks, Shelley. I never blow off sessions to have a glass of wine. We're in Australia, so. Okay, so what I'm going to do today is I'm going to try to focus the talk on the nucleosome as a direct component of epigenetic regulation during development, and specifically its role in compaction by the Polycomb-Group complexes. And to do that, I need to briefly introduce Polycomb. Many of you know about it. And the problem, the classical epigenetic problem of how you maintain body parts in an organism. This problem has been studied in flies initially, which is where the Polycomb-Group was discovered, and by Pam Lewis in 1947. And what Polycomb-Group is necessary for is maintaining repression. So if you want to get the body parts in the right place, you've got to get master regulatory genes such as ultrabithorax expressed in a spatially restricted manner. And early in embryogenesis as accomplished by sequence-specific DNA binding proteins. But later in embryogenesis, and notice this is an experiment of Jurg's when he was in junior high school or something. Later in embryogenesis, ultrabithorax is still spatially restricted, but it's held in a restricted pattern because it's repressed everywhere else by the Polycomb-Group. And if that goes wrong, all hell breaks loose. Here's a wild-type fly. This is work of Gary Struhl's. And here in the wild-type fly, ultrabithorax is spatially restricted. But if you have a homozygous mutation in the Polycomb-Group, you lose that spatial restriction, and now ultrabithorax is expressed throughout the embryo. This embryo is going to make intestine everywhere, including where its brain should be, which causes either death or being a Yankee fan, depending upon which is worse. Okay, so this is the classic problem in epigenetics, and that is you have a master regulatory gene. It's got to be on in some cells to specify a lineage, but it has to be held off in all the other cells so it doesn't make the wrong lineages in the wrong place. And the Polycomb-Group has to do with this maintained repression. And so how do you maintain repression, and how do you maintain it in a way that it's held in a maintained state throughout the life of the organism? So that is the original classical definition of an epigenetic event, the heritable phenotype, despite having two separate heritable phenotypes with the same DNA sequence. And so, as most of you know, there are two major complexes in the Polycomb family. There's PRC2, which has several components in it. Many people in the room were involved in discovering that this is a methylates lysine 27 of histone H3, trimethylates it. What I'm going to focus on today, however, is the PRC1 family of complexes. There are two flavors here. There's a canonical PRC1, which contains these four Polycomb-Group proteins, and I'm going to focus in mammals, and I'm going to focus on the CBX proteins, which contain the domain that binds this methyl mark, and I'm going to focus on polyhomeotic. These two proteins are not in the variant complexes that ubiquitinate, and so I'm going to stay focused on the canonical PRC1 complex and its role in maintenance. It is unclear what ubiquitination does in maintenance, again, from work from many people in this room showing that it might not be necessary. Wendy Bickmore and Jörg Müller in particular. Okay, so here is the model for what the canonical PRC1 complex does and how it's targeted. PRC2 takes the beads on a string structure, methylates it on lysine 27 of histone H3. That binding mark binds to the CBX protein. The key part of the first part of this talk is this protein and something else it can do, and that creates compacted nucleosomes, and I'm going to argue that this compaction process is critical for maintaining body fate in mice. That's going to be the first part of the talk. And so here's the outline for the talk. Here's the beads on a string structure. You can form local compaction. We know from scanning transmission electron microscopy that there are 4 nucleosomes per polycomb for PRC1 complex. I'm going to start talking about data we have that says that this is critical for vertebrate axial patterning. And then I'm going to move from there to talk about long-distance interactions, long-range interactions in compacted protein, and the role for polyhomeotic in establishing that. Okay, so what is compaction? What is this local compaction? So this is an experiment done by Nicole Francis in 2004 with Chris Woodcock, where we took nucleosomes, the beads on a string structure of nucleosomes, incubated them with PRC1, in this case fly PRC1, but the same thing happens with mammalian, and you get these compacted structures in which you have 4 nucleosomes being held in place by PRC1. So we looked at this and we thought, this is really cool. Maybe this is why chromatin is repressed, because how could a transcription machinery repress this? So that was one possibility, and the other possibility was that this was schmutz on a grid, and that it had nothing whatsoever to do with biology. So I'm hopefully going to convince you over the next few minutes that this is important to biology. One reason we think it's important to biology, and this is from a review from Danny's lab, is that a lot of proteins that are involved in repression do the same thing. They can all create these compacted structures on a grid. And we've been trying to do structural work with a lot of these, and we succeeded with the yeast protein SIR3, which makes these amorphous compacted structures on a grid. But those compacted structures, we can show using crystallography, form a very discrete structure in which the nucleosome is part. And as introduction to set up what I'm going to talk about with PRC1, I just want to remind everybody of work done 5 years ago by Karim Jean Armash, who's now at NYU, but was then a postdoctoral fellow in the laboratory, and also done in Dinesh Moazed's group, very similar structure, showing that the repressive protein SIR3 binds to the histone octamer. It binds to the proteins, the nucleosomal face of the histone octamer, very little to the DNA, and it creates basically a macromolecular structure that includes the nucleosome and the repressive protein. And so what this says is that repressing proteins can interact directly with the nucleosome in a very specific manner. And because of all the yeast genetics, we know that this is biologically relevant because we can explain about 30 different genetic mutations from the atomic contacts we see in this structure. What's important for what I'm about to tell you about PRC1 is how this structure forms. And it forms by an induced fit that we can see crystallography of a positive knob that is normally disordered on the SIR3 repressor protein. That positive knob interacts with the acidic patch on the face of the nucleosome octamer. And so this charge-charge interaction is a major driver of this structure, and this interaction with this acidic patch has been seen by Song Tan in numerous structures. And so that type of interaction is what's holding together the nucleosome. Here is the crystal packing unit where you see the nucleosome, a dimer of SIR3, another nucleosome, all packed together. And so there is a discrete structure to these blobs that we see. So what we would love to do to understand what PRC1 is doing is we would love to have a crystal structure of these blobs so that we would know exactly what amino acids were involved in them. We would mutate them. We would ask if they would have phenotype. And we and others have never been able to get a structure for this. But the other thing we can do is figure out what amino acids are involved in solution studies and mutate them, and I'll show you what happens when we do that. So in order to do that in mice, so what I'm going to be talking about the mammalian PRC1 complex throughout this, in order to understand whether or not PRC1 compaction is involved in development in mice, we had to determine where the amino acids were in this very large complex that creates this compacted structure. So PRC1 is very complex. There are multiple different subunits that can all interchange with each other. And we ended up finding out that CBX2 is the main subunit that is able to compact most strongly, and that there is a positively charged disordered region within CBX2 that is responsible for compaction. Dan Grau, who did this work in the lab, was able to identify a similar domain in PRC1 components from all of these organisms: flies, mice, worms, frogs, and zebrafish. This indicates that PRC1 from all of these organisms has the capability of forming these compacted structures, arguing that it's important to repression, and in particular defined the domain in CBX2 that was responsible. It's, again, a disordered domain with a lot of lysines and arginines, and if we make the lysine and arginine to alanine mutations, 23 or 13 KRA, these proteins are no longer able to compact. So the prediction is, if this is actually important for function in vivo, if we make mice that knock in these mutations, we'll actually see developmental phenotypes on axial patterning. And so I'm about to show you those data. Here's the wild-type protein. Here is the 13 amino acid mutation. Here is a solution assay for compaction. The wild-type protein is very active. The 13 amino acid mutation, called 13 KRA, throughout the rest of this talk, is intermediate. 23 KRA is significantly impaired for compaction. What happens if we put these into mice? So Mei Sheng put them into mice using standard, first CRISPR standard and then CRISPR targeted. We have mice that are homozygous for these mutations and that are expressing completely appropriate levels of CBX2. What sorts of phenotypes can we look for? Well, mice aren't like flies. There's so much redundancy in the system. Remember all those homologs of CBXs that I showed you. So the phenotypes aren't anywhere near as dramatic as they are in flies for Polycomb mutations, but they're well known in the literature, and the CBX2 null allele is well known, and it does anterior-posterior transformations of the axial patterning, C7 to T1 and others, and they consist of things like extra bones, like this extra bone is the phenotype. So if compaction is important, these mice should have disrupted axial patterning. And they do. And so this is the 23 KRA homozygous mutation, and it has 5 different anterior-posterior transformations in axial patterning, and they phenocopy what was seen in the null, except in the one case where the original paper didn't look at it. So the 23 KRA mutation is a loss-of-function mutation that phenocopies axial defects of the null. So this supports compaction being critical for development. What about the 13 KRA? Remember, this can compact a little bit. It's intermediate between the wild type and the 23 KRA. And what you can see is it also has some transformations. It doesn't transform as many places as the 23 KRA or as the null, so it has a decreased function in axial patterning. So now I'm going to show you what these phenotypes actually look like, to show you that in addition to the number of segments that are altered in each of these mutations, there's actually a visual difference in what types of mutations you get. Okay, so here's where you have to look at axial patterning. You're looking at the C7T1 transition. This is what C7 normally looks like. This is all work we did with Cliff Tabin's group and Matt Schwartz in Cliff's lab. So here's wild type. Here's what C7 normally looks like. T1 has extra bones coming off of it. In the 23 KRA, you see very clear extra bones coming off of C7. They're ectopic ribs at C7. The 23 KRA shows these long exterior bones. The 13 KRA, the intermediate mutation, shows these bones, but they're much shorter. So in other words, not only are we seeing different numbers of changes, but we're also seeing a different phenotype visually between these, where the thing that cannot compact at all has much stronger phenotypes than something that can compact to an intermediate degree. I'm going to show you one other segment, L6 and S1. This is what L6 normally looks like. The anterior posterior is going to make L6 look like S1. This is what S1 normally looks like. It's got these little wingy bones coming off of it. The 13 KRA will have a wingy bone, but usually only a unilateral on one side. The 23 KRA will have a complete transformation to the full bilateral axial change. And so the key findings, there's a very clear finding here, and that is that if we eliminate the ability of CBX2 to drive this compaction with point mutations, we have two separate strains, and the mice show a strong correlation where if you mutate that ability, you get the axial patterning defects. So you're all probably wondering about controls here. How do we know this protein is actually still doing everything it's supposed to do? Is this really a specific mutation that's only impacting compaction, or is it impacting other aspects of the polycomb process? And so we can examine that molecularly by looking at these mutations. You can't do that in the cells that do this here because there aren't enough of them, but we can do it in ES cells, and we can show in ES cells that this mutation has a molecular phenotype. Here's wild type protein on a polycomb target, 3 different lines of the wild type protein, or a control mutation showing the repression that one normally gets with, here's without the protein, with the protein, without the protein, with the protein, you're seeing repression. The KRA mutations are no longer repressed, and so we have a molecular phenotype, and we can ask what this protein is doing in the cell and what the rest of the Polycomb-Group system is doing in the cell in these mutant cells. And what we see is that K27 methylation and H2A ubiquitination are not altered by ChIP-seq. We see CBX7 and ring 1B binding patterns are not altered by ChIP. And so we know that the whole system, the whole rest of the system is intact. And CBX2 is still functional in other characteristics. It still forms the full PRC1 complex. But the most important observation that we made is that there are other phenotypes of the CBX2 null, and those other phenotypes are not impacted by these mutations that disrupt compaction. There's birth weight, sterility, and sex reversal phenotypes of the CBX2 null. We see none of those phenotypes in these mutations. And so that says that the KRI mutation is a specific is a hypomorph that impacts axial patterning and not the other functions that CBX2 does. And so that says that CBX2 is maintaining a lot of function. Finally, what about targeting? Does it go to the right places? That's actually the hardest thing to address. It's really hard to figure out what's exactly going on because the mutant, remember, is mutating all the lysines. And so if you mutate a whole bunch of lysines, it's hard to do ChIP because ChIP is cross-linking, which goes through lysines. And so that means that if you mutate the lysines, you don't expect to see very much ChIP signal. And so we get ChIP signal, and we see smaller amounts of ChIP signal. We're mutating 60% of the lysines, and we see 60% of the signal, which is probably actually just sort of random that they line up. And so that's consistent. But where it goes is very similar for the wild type and the mutant. So targeting appears normal. Okay, so what the conclusion from this part of the talk is that mutations that impair compaction specifically generate defects in axial patterning and not in other functions of CBX2, and that the ability to create these compacted structures, when you impair it, you get specific axial patterning defects. And we conclude that in vitro compaction ability is necessary for epigenetic maintenance in vivo. That's what we believe is happening here. Okay, so that was the first part of the story, and that says that whatever this compaction process is happening and appears to be important for appropriate developmental transition. And so what I'm going to spend the last 9 minutes and 6 seconds of the talk getting into is long-distance interactions in compacted chromatin. And I'm going to argue that what happens with Polycomb, the way you get a repressed domain, is that you take these locally compacted domains and you organize them into a very specialized long-range compacted structure, and that this compacted structure is critical for repression. And I'm going to present the hypothesis that this structure is actually a key part of epigenetic memory, that once you get everything into this structure, everything is just going to be in a situation where it's going to get remembered. Okay, so does PRC1 also impact higher order structures in addition to these local interactions? Can it create compacted domains on chromosomes? And this is the work of Sharmisa Kundu, and Sharmisa is here and is presenting a poster on Friday, which will give much more detail. I'm only going to show a couple experiments here. Sharmisa emailed me her poster number 10 minutes before I started talking, which indicates that she thinks I might actually remember it now, which is very nice of her. This is work with Job Decker and Gaurav Jain and Jeannie Lee and Hongjae Sunwoo, who did the 5C in the imaging, and critical work in bioinformatics by Ruslan and Fei, who figured out how to interpret all this, and Ruslan is also here. Here is the system we use to figure out if there's long-range compaction going on. We looked at Hox clusters, we looked at PRC1, and we looked at the Hox-A cluster in embryonic stem cells and in neuronal precursor cells. And that is because PRC1 is spread throughout the Hox cluster and represses the whole Hox cluster. In ESCs and in neuronal precursors, it is only on the right half of the Hox cluster, and this is ChIP-seq showing where they are. Does this chromatin organization change upon differentiation? In order to do this, we use 5C. So most of you probably are familiar with the C technology. If two fragments that are in different places are actually physically, spatially near each other, they get ligated. What 5C does that is different from the other C technologies is you use a whole bunch of primers, and so you're amplifying a whole bunch of primers together, and it gives you very high resolution, but only over the region of the genome where you have the primers. So we're able to get very high resolution, 2 or 3 kilobase resolution over 10 megabases, which is what allows us to see some fine scale structures. And what Sharmisa looked at using this is Hox regions and other regions that are repressed by PRC1. And what you can see here is the 5C map, and you can see the TAD domains that span Hox-A, TAD, TAD, TAD. And what you can see on top of that TAD structure is a structure here, which is not a TAD structure. It's smaller than a TAD structure that is forming this square of interactions directly over the Hox-A gene cluster. That structure right there goes away in neural precursor cells. Now we're activating this part of the locus. That large square of interactions goes to a small square, and now you start seeing these strong interactions. This is a Z-score map for people who do this. We can see these on the primary maps too. This is showing that you can see interactions out to the enhancers in this HOX-A cluster. So that's the first major conclusion, is that with this fine-scale 5C mapping, we're seeing a restricted domain that maps only over the HOX-A cluster and becomes smaller as the HOX-A cluster becomes less occupied. Can we see this in microscopy? We use oligo painting, technique developed by Brian Beliveau and Ting Wu, which puts a probe every kb throughout the 100 kbs. And what we see here then is we can see the entirety of the HOX-A region, and we see compacted structures, as Wendy Bickmore first showed with using 2 sites to look at these things, and we see the whole thing getting held together. In neuronal precursor cells, where it opens up by 5C, it also opens up by this storm imaging. Is this canonical PRC1 or variant PRC1 that's doing this? Sharmisa analyzed that by using CRISPR to knock out proteins. She knocked out the polyhomeotic gene and asked what happened to these domains. So here is the small domain that is formed over the HOX locus, HOX-A. If you knock out polyhomeotic, you completely remove that domain. Here's a difference map. And you also increase the positive interactions. You reactivate the locus and increase the positive interactions going out to the enhancers. And you get this difference map that shows that you've made a clear difference in 5C interaction as you knock out polyhomeotic. And we can see this by storm imaging too. This again is work of Hongjae's. Here's HOX-A and HOX-B, both showing the repressed domains. This is the locus that Wendy was looking at in her original work on this. And here we see the pH knockouts and showing that we're expanding these structures into much larger and decompacted structures by knocking out polyhomeotic. What about variant PRC1? Is it doing this? And so we got from Wendy Bickmore's group very nicely their ring 1B 153A ESC cell line. They had previously shown that this does not impact compaction in their assays. We also saw very little impact of compaction in our assays. Here's the wild type. Here is the 153A mutation. You can still see this small square, and this is the difference map showing that there isn't very much difference. And that differs from the ring 1B knockout, where you completely get rid of this interaction and you see a big difference. So ubiquitination is not required. So I'll finish this part of the talk, and again, there's more data that's on Sharmisa's poster. I think it's 245B. One of those letters or numbers is probably wrong. And you can look at these maps by drawing a line between everything that interacts with each other. And what you see very clearly when you do that is from this 5C analysis, the fragments that interact with each other are pretty much directly under where we see by chip binding of PRC1. So we believe these are small domains and that their boundaries are determined by where PRC1 binds. One way of testing that is to look at parts of the genome where there are several Polycomb repressed genes in the same neighborhood, but where the binding is more spread out. And we actually have that data because Sharmisa planned very well and looked at gene sets like that. And here is a set of 3 genes, NKX24, 22, and PAX1, clear binding of PRC1. And now by 5C, you see interaction only between the fragments that are bound by PRC1. All these little colored rectangles in here are fragments that we could see by 5C, but we're not seeing, and they are also where there is no PRC1 bound. And so what we believe is happening is that PRC1 is forming a domain over genes, and then those domains are linked together, and the HOX clusters are special because PRC1 is bound everywhere and is creating a whole balled up region. Finally, these domains are different than TADs. These PRC1 domains are much smaller than TADs, and the boundaries do not correlate. TAD boundaries correlate, as many people have shown, including many in this room, with CTCF, cohesin, and mediator. Here is showing the TAD boundaries and the probability that they'll be near one of these three, and it's much above average in the genome, whereas these boundaries are average in the genome. And so to conclude this part of the talk, what we've identified is PRC1 domains and the fact that they are much smaller organized domains than TADs. They occur where PRC1 is bound. And we believe that what happens is you get this low local compaction, and this local compaction goes to these compacted domains. And my time is up, and I'm done, so I'll simply finish by saying that two types of compaction are necessary for proper memory, for actually proper repression during vertebrates. The hypothesis is that they're also involved in the memory. So what we know is we know that compaction, that if you mutate the domains necessary to make this compaction, you get you disrupt axial patterning. This follows work that Jörg Müller and Ting Wu and I did in flies many years ago with the posterior sex comb proteins. And that the polyhomeotic protein part of PRC1 is necessary for these bigger domains. And it is known that mutation of the same organizational domain of PH disrupts these domains from Nicole Francis and Kim's group. And Kosecki has done some very nice, Kosecki has done some very nice work to show that that also creates these axial patterning problems. So both of these steps are necessary for axial patterning. And the hypothesis is that what they're also necessary for is the memory. And I'll close by saying that the hypothesis we'd love to test is that these domains are what are remembered. These domains are either separated spatially or in solution, so that when they form, they are automatically reformed as cells replicate and cells divide. And therefore, that if you really want to get the whole thing to work, you need all of these steps. It's well known that methylation of K27 is necessary for full repression. And I'm arguing that you also need to compact nucleosomes with the CBX2 component of PRC1 and compact domains with pH, and that all forms together to create a structure which is not only the engine of repression, but also the engine of memory, the last being the hypothesis. This is work from Dan Grau, Joe Garlick, and Karim Jean Armash on the original work on the compaction domain. Mei-Sheng Lao did all of the mouse studies. Sharmis Lakundu did all of the compaction studies. A lot of help from Cliff Tabin's group, Jeannie Lee's group. Ruslan did a lot of beautiful bioinformatics, and all of this was started by Nicole Francis and Karim Jean Armash when they were postdocs in the lab. Thanks.
Where are our volunteer undergrads, graduates?
Somebody in
Great talk. So you mentioned somewhere in the middle that the PRC1 associated domains are different from TADs and that they don't co-localize with CTCF, cohesin, etc. So, but the Hox locus that you used to demonstrate all these things has a bunch of CTCF sites, as shown by Danny Reinberg. So how do you reconcile what he said?
Right, so there are CTFs, there's a CTCF site, for example, right in the middle of HOX-A. And so what our hypothesis is, what fits with the data, is it's a CTCF site right in the middle of HOX-A is critical for that TAD boundary that's forming there. And that TAD boundary is critical, and Denis Deboul has data on this. Danny has data on this. They can argue with me if they think what I'm saying is incorrect. But that boundary is important for reaching out towards enhancer regions that are outside of that is going to turn on those genes. And so what we're arguing is that what PRC1 is doing is creating a separate domain on top of that when the whole thing is repressed, that is basically pulling that CTCF site out of the ability to form that bigger TAD domain boundary, and that you're getting a smaller localized compacted structure that now can no longer reach out to enhancers and now no longer become activated. That's what our hypothesis would be. Did that answer your question? Yeah.
So given that these repressed domains are loaded with serine 5 phosphorylated polymerase 2, what model do you have in mind for how the compaction prevents that polymerase entering productive elongation?
Yeah, that's a great question. So what polymerase is in there? What is that polymerase doing in there? What else is in these domains? So I think that there is very likely to be much more in these domains than Polycomb-Group proteins in the DNA. I would like to think that there are long non-coding RNAs in there. That's one of the things we're working on, and that they are involved in some way in these domains. And polymerase is expected to be in there, and what is its actual nature and what is its role in the whole process.
Hi, Bob. Great talk. Thanks. I was just wondering, how do the local domains find each other to do the more overarching structure? Is it just whoever is nearby in the smaller region, or do you think there's some finding mechanism?
Yeah, it's a great question. And so the question is, we're with the compaction domains in the microscope that we're making structures with 4 nucleosomes and 1 PRC1. And are those self-organizing, or are those being organized? What would happen if there were no pH? And what's interesting there is, I didn't get into this, but one of the domains that Sharmisa's looked at on PAC6 isn't actually very impacted by deletion of pH. And so it's perhaps if you're small enough, you just need that local compaction to hold things together.
I'm wondering about the nature of this nucleosome compaction. Is it, do you think it's the linker gets shortened and then the nucleosome get closer together, or the linker length maintain the same, but the nucleosome get pulled together and secrete the linker? In that case, the transcription factor can still bind to linker DNA, right?
Right, and so you're asking 2 separate questions. One is the compaction actually affecting linker length in the cell? And we don't have any data on that, and I don't know if anybody in the room does have any data on that, so I don't know the answer to that question. And the second part of the question you're asking is, we know that in vitro that linker is still available even when those things are compacted, right? And so is the linker DNA still available on the outside of those compacted structures or not? And if it is available, what is it available to? And that gets towards Wendy's question, which is basically, are you repressing because you're preventing the DNA from being accessed, or are you repressing because you're creating some sort of macromolecular structure that is impenetrable just for stuff getting in that needs to get in to turn things on? And I would put my 6-pack of beer on it being an actual big structure that is just impenetrable to all the right regulatory factors getting in, but that's something that obviously needs to be tested. That's hypothetical.
Okay, I think we'll go on. Bob, thanks for giving us a great start to the meeting. Wonderful talk.